Short week, just today and Wednesday. Let's make Wednesday a coffee day. So we'll do coffee Wednesday. No, Easter. Good Friday on Friday.
News to me, wow.
Is there Monday?
No, there's nothing Monday either.
Oh, okay.
Thanks to Grace. Grace reminded me of all those things at the beginning of the semester. So what we will plan to do is on Wednesday we'll start Ebola. We're not going to talk so much about the virus itself. We're rather going to talk about the epidemiology of the infection. So we'll talk about, basically I'm going to kind of do like one mini lecture on all things related to epidemiology. So how do we characterize different types of outbreaks based on location? How do we characterize disease and symptoms of an illness? And then we'll spend some time with that and discuss particularly the 2014-2015 Ebola outbreak. And then on Wednesday next week, we'll get into discussing Ebola virus in general, focusing primarily on structure and everything up to biosynthesis. And then we'll cover biosynthesis after the exam next week on Friday. My goals over break are to write the take-home so that I can get that to you as soon as possible, and then the in-class portion is not this Friday, of course, but next week on Friday. Okay, so that's a little update on where we're at. I emailed UW to find out about sequences. I didn't assume I would hear anything before this morning, so I'll let you know once I know. I think some of you may have seen the email. I apologize that this is going to be a little condensed, but I am taking that into account then when we talk about, okay, so what are the final results on the project, okay? Once we get those sequences in, I'd like to do a couple of things. One, we're going to schedule a day in class after you've had maybe a couple of days with the material, with the sequences, so that you can use that time as a work period. And I'll be, of course, here to help out with that, and I'd like to kind of touch base with each group to talk about the project and to see what questions you have just as you're getting started with the analysis. And then I do want to schedule kind of like a half-hour meeting with each group outside of class time within a week following that or so, just to touch base and see where you're at with progress, okay? So those are things that are coming up. We're over halfway done with the semester now. Yay! All right. So we left off on Friday talking about different HPV-caused cancers and talking about some of the characteristics of transformed cells, which we can kind of consider to be like cancer cells in vitro or in a petri dish. But not all transformed cells are going to cause cancer. And the reason for that is cancer cells oftentimes also have to have a couple of additional characteristics like avoid surveillance from the immune system, be able to promote angiogenesis. And those are characteristics that not all transformed cells are going to have, okay? So I just wanted to kind of reiterate that real quick. I suppose what I want to start out with today is just, the goal today is just kind of to do review of material. We've covered most of everything at this point. So I wanted to start out with, are there any questions on what we talked about on Friday or anything else with the HPV life cycle that's still kind of foggy or unclear right now? I know I'm putting you on the spot, but... And if not, that's fine. I'm sure they'll come up as we go along. I think we left off with this image, there we go, that image there. And this is just, again, this is an immunofluorescence looking at stratified epithelial cells. The dotted line represents the basal membrane. And in this image, they're looking at HPV 16 infection versus an HPV 11 infection. And the red represents MCM2, which again is an indicator of cell replication. The E4 protein from HPV is expressed in those differentiated keratinocytes up here towards the top. And again, E4's function is to modify the filaments to allow for the release of the virus from those keratinocytes. And then the blue represents just DNA. So one of the things that we pointed out here that is a key characteristic between the high risk, which includes 16, so that's our high risk, and those are the ones that we associate with cancer. And a difference between those and the low risk is that we have much more proliferation occurring in those undifferentiated epithelial cells than we find within the low risk ones. And we think that that contributes to the cancer that is caused by those high risk. Sabrina.
So the white dotted line is basal?
That's our basal membrane, yeah. So below that would be, now you're getting into the dermis and out of the epidermis. The other thing, and Shania had kind of asked this question about, well, how we were talking about telomerase or the telomere. I can't even say it the other way. Anyway, we were talking about the ability of E6 and E7 to turn on that enzyme and to maintain the length of those telomeres so that we have these cells continuing to proliferate even though they may be differentiated. And this enzyme is one that typically is expressed in stem cells or undifferentiated cells to ensure that the length of those telomeres remains long. And we said that a cell goes through a certain number of replications before these telomeres are so shortened that the cell no longer replicates. Well, to maintain their length, that enzyme's got to be expressed. And E6 and E7 help to control that by, we said that there was two ways. E7's ability to bind to E2F and drive transcription will allow for expression of telomerase. And E6AP with E6 together will bind to a repressor of telomerase. So in conjunction, E6 and E7 are going to promote the transcription and ultimately translation of telomerase, allowing for the maintenance of those, of the genomes and maintenance of those telomere lengths. Sabrina.
Is the E6AP the same mechanism that degrades the P53?
Yeah, so here, it absolutely. So it's going to be adding multiple ubiquitins to the NFX1 protein and therefore leading to its degradation so that you lose its repressive function. Yeah, absolutely. Grace, did you have a question?
No, I figured it out.
Okay. Let's start with this. This is one that I didn't include on the sheets that I gave you guys, but I think this is a pretty good question. So, in thinking about cancers, one of the things that we want that we're kind of at a point right now in our history of cervical cancer, head and neck cancers, etc., others that are associated with HPV, is we're at a point where we're starting to now get data back on how effective are these. So the HPV vaccine's been out for about 15-ish years, roughly. The original phase 3 clinical trials were probably done about 20 years now, so we're actually finally getting back some really good data to determine, do these have an effect in inhibiting the formation of cervical cancers and other forms of cancers. So the question becomes, what kind of data would you want to look for to indicate that preventative therapies like a vaccination actually work? What would you look for? This question may be a little bit easier to answer now than it was 10 years ago. Go ahead, Jonah.
Look at two groups of people, like who have gotten the vaccine and people who haven't, and see like what percentage have contracted HPV and what percent haven't.
Yeah. So, and that has, or that's kind of where we're at now. So have a group that's given a placebo or a sham, have the other group be given the vaccine. You give them the same exact information about sexually transmitted infections, HPV, and the ability of it to cause cervical cancer and other forms of cancer. You let them live out their life, and then you follow up with them every so many years to look for the formation of cervical cancer. And in those experiments, what we're finding is those that receive the vaccine, it's about 80-90% effective or more. If we break it down to look at what percentage of those cancers are being caused by HPV 16 or 18 versus other high-risk or low-risk cervical cancers, the vaccine works in about 99.7% of the cases of cancers caused by HPV 16 and 18. So it's very effective against the strains that it's been designed to target. The story was a little bit different maybe 15 years ago when we were just starting to look at the results. One of the things is it's not as if all of a sudden cervical cancer shows up. There's a progression. And this is true for all cancers. And so in HPV world, the different types of, and this is what they actually do with pap smears as well, they will grade the different lesions based on how aggressive they are and what they look like. So there are levels known as SIN 1 through, I think it's through 4, 1 being the least and 4 being the worst. And SIN, I cannot remember, it stands for cervical in something, and I cannot remember off the top of my head what it represents, but it is basically a measurement of how aggressive or how, what do the pap smears look like. The more aggressive the form, the more abnormal cells are, that gives a higher grade. And so there's a progression. SIN 1 represents kind of like the lowest of precancerous lesions. SIN 4 are the worst of those precancerous lesions, and then you have cervical cancer. And so maybe 10 years ago, actually, when I was in graduate school, this data was coming out. They were looking at data determining, okay, so what is the progression of SIN 1 or SIN 2 in individuals who had received the vaccine versus those that had not received the vaccine? Those individuals were probably still too young to have developed cervical cancer at that point. It takes about 20 years or so from an HPV infection, HPV 16 or 18 infection for cervical cancer to develop. So they were looking at these precancerous lesions, and what they found was these lesions were much less frequent in individuals who had received the vaccine than those that hadn't. Again, arguing that it was preventing the progression of cancer. Okay. What else could we look for? If we wanted to type a cervical cancer by what form of HPV is causing it, what might we do?
I mean, I don't know how you do this, but look at the replication of the, like how much replication there is.
Okay, we could do that. But one thing to recall, I'm going to back up real quick. Remember that in the actual cervical cancers, once these get formed, there's only a few things in there, right? It's E6, E7, maybe part of the LCR. So the replication at this point isn't going to be viral replication per se. The viral genome is going to replicate when the host does, just because it's integrated into the genome at that point. Well, what else could we do? What other DNA techniques could we use?
Sequencing?
That'd be the easiest one. PCR and then sequence. These different strains, the E6 and E7 from HPV 16 are going to look different than the E6 and E7 from HPV 33 or one of the other strains. So we might be able to type just based on sequence comparison. You could look to see if the LCR is there, and you might assume that there are slight differences in the LCR as well. Okay, so just by sequencing, we might be able to place it into a particular category or strain class. And that's what's being done to determine which forms of HPV are contributing to these cervical cancers. Sabrina.
When E6 and E7 was incorporated into the genome, is that along with viruses?
So it would just be, at this point, no more viruses being made.
So then is it still like able to be transferred?
Ah, that's a great question. No. No, it's not. Now, there's always a possibility that you have maybe a subset of cells in which this integration has occurred, and if the infection isn't being treated, then you might still have some cells that are producing like viral particles. But at this point, these cells that have the integrated genome would not be able to transmit it. This kind of brings up, this brings up an important issue. I mean, we're talking about preventative measures here. There are other individuals, other groups. There's one researcher at UW, for example, who's focused not so much on the preventative, but the therapeutics. How do you prevent the spread of HPV once somebody has an HPV infection? So Sabrina, you could argue a point where, like, if somebody has cervical cancer or has genital warts, you want to put them onto some sort of therapy or treatment in order to block the replication from occurring. We don't really have great methods for that right now. And part of the reason why is that HPV's life cycle is so intertwined with the host cell. It uses the host cell DNA polymerase. It uses the RNA polymerase. So you'd have to talk about blocking very specific proteins in order to block that process. E4, maybe, for example. One last thing. Jonah, you asked a question about NK cells a week ago. Why don't NK cells recognize us? I think I figured out the answer, but I've got to read my notes to fit. Okay, so what I found, there's a couple of answers, but one of the things that we said is E5 is the one that will disrupt MHC1, and that's what NK cells recognize. Apparently there are 4 or 5 different types of MHC1, and so what E5 is repressing are those ones that are recognized by CD8 positive T cells, but not those that are recognized by NK cells. So in other words, overall, if you look at it as a whole, it's repressing the amount of MHC1 on the cell surface, but it's only a subset of them that it's repressing. So it would reduce the amount of detection by CD8 positive T cells, reduce the killing by that mechanism. And then also, because it's not repressing those that are recognized by NK cells, blocks that mechanism as well. Yes, it is. No problem. All right. I'm going to... Oh, go ahead. Paige, absolutely.
So, because men can't really be tested for HPV, how do they...
Yeah, you're right. Not in the same way that females are with a pap smear. Yeah.
Right. How do they know if the cancer, like penile cancer or something, can be tested for HPV? Why can't they find a way to test men for HPV without cancer?
darn good question. I mean, usually there's a physical symptom. No, I take that back. I was going to say there's a physical symptom associated with that, but that's usually with the low risk. You have some sort of genital warts.
Yeah.
So,
But for like the high risk,
but for the high risk, man, that's a good question. I don't know the answer. The only thing I can think of, God bless that I'm talking about this. I don't know if a male is going to say, okay, you can take a sample of my penis and test it for HPV, right? Females are the superior species, and there are numbers of reasons why. I don't know. I don't have a great answer for that. I mean, I guess because the medical field is male dominated, and I don't know. I don't have a good answer. It's really a great question, though. Yeah, I have no clue. I do not have a good answer for it. Damn, that's a good question. That's a really good question, Paige. Grace, then Jonah.
Well, mine's a totally different question. So
Is yours similar?
A little bit.
Okay, go ahead.
Does your body ever make antibodies for HPV?
Yes.
So why couldn't you just test for antibodies?
That actually would be a good mechanism.
Does that exist right now? Or?
Good question for you to investigate. I do not know. I think what you would have to consider is what are those antibodies going to be targeted against, right? Part of the reason why it may be tough is in order for antibodies to be produced, the mechanism is you've got to have free-floating HPV virions that are engulfed and then presented on the surface of macrophages. Those then have to interact with CD positive or CD4 positive T cells, so the T helper. They would stimulate the B cells to produce the antibodies. So you have to have, in order for this to happen, you have to have a mechanism by which HPV viral particles are being released. That's not happening in the cancer per se.
How would those virons be released, like, not outside of the cell?
That is
Or, like,
another really
outside
good question, and I don't know the
So
answer to
I don't
that.
know how it would,
I have to imagine that the B cell response to HPV is really weak. What you'd really want to do is train CD positive or CD8 positive T cells, so the T cytotoxic and the NK cells, to recognize those tumors. And there are folks that are working on those types of mechanisms for other viral cause cancers and for non-viral cause cancers as well. Yeah, really good question. Grace.
So I was just gonna ask, is BRD4, is that like encoded with the virus, or is that
That's
like,
a host protein.
That's a host protein?
Yeah.
So is there a way that you could like, I don't know.
You might be able to create some sort of molecule that would inhibit the ability of BRD4 from binding to E2, right? So you could do, and there are a number of ways to do this, right? We've talked about, I guess, if a virus is free-floating, neutralizing antibodies work really well to inhibit infection. And actually, we'll see that with Ebola. If for something that's endogenous, that is inside the host, and like in the case of HPV, E6 and E7 is integrated into the, I mean, it wouldn't matter in terms of the cervical cancer, but in order to prevent the, I guess, prevent the spread of the virus, that's where that mechanism would be really beneficial. You'd want a small molecule that can get into the cell to inhibit that interaction. And so UW has what's known as the small molecule screening facility, and it is a bank of hundreds of thousands of chemicals that have been produced by different chemical companies, and they're just libraries. So to do that experiment, what you would do is you'd take your fibroblasts, you'd put in probably E2 and a plasmid that E2 can bind, and then look for, when you add these different chemicals, do any of them inhibit the inhibit or reduce the amount of viral particles from one round of replication to the next. Of course, you don't want it to kill the host cell because that's very bad. But those screening processes are done. And out of those hundreds of thousands, you might find like 50 to follow up on. Yeah. So that is possible to do. It's expensive. Nice thing is it's largely automated nowadays. So like if you have a fluorescent plasma, a plasma that produces like a fluorescent protein or something like that, it can make the experiments much easier to do. Annie, did you have a question?
It was kind of about the antibody thing.
Yeah.
So, making sure I understand the life cycle of HPV.
Yeah.
It's really only,
Go ahead.
yeah, so it's really only outside of the cells at the beginning and the very end, right? So like at the beginning it infects, but then the entire way to the surface it's just being replicated with the cells, right? So there's no virus.
Yeah, you don't really have that exit until the end when it's on the surface.
So like the antibody would have to be attacking it before it would get, which it wouldn't.
Which it wouldn't necessarily unless, that's where I see an antibody mechanism being kind of hard to do. Unless, no, that wouldn't even work. I take that back. I think an antibody mechanism would be really weak here.
I don't know much about antibodies. I'm gonna
Either do
preface that. And I don't know much about immunology.
Either do I, right, Paige?
No, I don't want to sound too So, I don't know, I know that you can attack sick cells, but they're not antibodies against the cells that would have the...
No, it would be...
against the particle.
Yeah, so like the antibody would be against a viral, it would be against some foreign protein or an epitope of a protein. Usually when you're attacking sick cells, that's going to be NK cells or it's going to be CD8 positive T cells that are attacking that. So that mechanism is usually antibody independent.
Okay. And the cells aren't like observably sick to the body, like.
I don't think so. Yeah, that's a good question. I don't think so. Now, it might have presented on its surface some viral proteins, which would be recognized as foreign and could be considered to be signals of sickness. Mm-hmm. But like we said, in the case of E5, it represses that the receptor, which is called MHC2, or MHC1, sorry, that presents those foreign particles. So by repressing that, you would repress the ability of the immune system to recognize those cells. And also, this is kind of this is an area under low immunosurveillance. There's not many, you don't have high concentrations of CD8 positive T cells or NK cells in here. So because of that, it may not be well detected. That would be my guess. Jonah?
So like if an HPV vaccine is like just L1 or whatever it is.
Yeah, great question.
How does that protect against like just a viral particle getting in there and just infecting it?
Okay, that we can address with antibodies. Antibodies are really beneficial in neutralizing the initial steps of the infection. But of course, in the course of a regular HPV infection, by the time that antibodies would be produced, the virus is already hiding inside the host cells.
So those antibodies just like chilling everywhere? And
Yeah, so you would want to, in all reality, our body or our B cells will produce different types of antibodies dependent on different cellular signals that contribute to B cell proliferation. So the major one that we talk about is IgG. This is kind of like the free-floating, the typical Y-shaped antibody that we think about. Those we can call those neutralizing antibodies. They would bind to L1, prevent L1 from binding to heparin sulfate or the other receptors, and therefore block the attachment and entry in the first place. You might also hope that it's producing IgA. This is the antibody that's found in mucosal linings. So in areas where HPV, at least the sexually transmitted forms, those high-risk ones, are going to be most prevalent, are going to be where mucous membranes are. The vagina, larynx, the penis, those areas are going to have mucosal membranes associated with them. And so having a high abundance of IgA there would, again, neutralize the infection from occurring. Yeah. Paige.
How, it's like, a lot of the high-risk ones besides the 16 and 18 can clear themselves,
Yes.
and then can you be reinfected with those since the antibodies aren't that?
You might. I don't know. I don't know if anybody's really investigated that all that much in depth. I would imagine that if it clears itself, that means that your body has developed an immune response to help clear it. And so my guess would be that's just a form of natural immunity at that point, that you may have the antibodies to protect against it. I don't know,
Because how
though.
else would it clear itself? You know?
Yeah, that's a good question. It wouldn't unless there was an immune response, right? Now, one of the things we don't know about is how long-term are these effects of, for example, Gardasil and some of those other HPV vaccines. We don't know if they last, if they generate an immunological memory that lasts for 20 or 30 years. We have no clue. I mean, we typically are, the design behind them is for them to work in a period of time when somebody might be expected to be most sexually promiscuous in their life. That's the design behind them, is to start somebody on these vaccines before the first time they have sex, and hopefully that it works as long as the time that they have multiple partners, if they do in the first place. That's the idea. Sabrina.
This is kind of unrelated, but, so those are all like external ways to control that. So is the only internal way that, like, self-surveillance itself is the preventing apoptosis? That's the only way.
Yeah, yeah, and then promoting cell proliferation, but that's not necessarily a way to, I mean, the proliferation is going to help to ensure that there's an expansion of virus, right? But the prevention of cellular death ensures that the virus will be maintained, at least. Yeah. This is a really good conversation. I'm glad we took the time to do this. Are there other questions on this topic? One of the things to bring up, I was looking over the quick sidetrack, sorry. I was looking over our schedule, and you know or may remember that the last week is kind of set aside for you guys to explore something with virology. In the past, it's been, you describe to me a virus that we haven't talked about, describe its life cycle, etc. I'm thinking about reformulating this. And so, Jonah, your question about, I can't remember what it was, where I said for you to look it up, like 10 minutes ago. Whatever it was. But if there are more tailored or specific questions that you guys want to investigate, I'm totally on board for that. So, like, let's say you want to look at a different virally caused cancer. I think you should have the ability to look at that if you want to. So start thinking about what you might want to talk about. This is kind of a free assignment for you to just show what you know at the end of the semester. We'll talk about it more later on, but, you know, start jotting down some ideas if you have them. Okay. So a couple of quick things. One, I'm a nerd. We all know that. So, somebody a few years ago created these playing cards of viruses, okay? This is out of California. So, here's the playing card for HPV 16. With the icosahedral structure, two different images of the protein structure of the capsid. So, this is a virus trading card. It's part of the alpha papillomavirus 9. The genus is alpha papillomavirus, family Papillomaviridae. Tells you it's double-stranded DNA, icosahedral, and then there's some information on the lower left for you as well. I think we should make these for our bacteriophages. Side note. We all know it. I mean,
Do you have these cards?
no, because nobody actually like printed them. They're just available online. So, yes, I have them saved on my computer, but it's like a business card, right? All right, let's do a review question or two. So I gave you guys a sheet. Did anybody not get one of these when you came in? So here are just a couple of them. These ones come directly from, well, at least the top two come, or sorry, the middle one comes from the questions that I put for you guys already up on Blackboard. The first and third are other ones that I kind of thought about that might be interesting to look at. So for example, the top one is find another virus that induces transformation or cancer, identify genes from it that contribute to it. The K-cyclin from KSHV would be one of those examples, okay? I think we'll skip this one for now, but this could be a potential topic for somebody if you wanted to use it as a potential guiding point for the final assignment on the semester, okay? The last one, actually, let's do the last one, and then we'll come back to the second one with whatever time we have left. So the last one reads, transfection of HPV positive cells. So let's just assume this is a general HPV infection. Okay, so we've infected with HPV, now we're transfecting in poly-IC. And you may remember we saw poly-IC on the take-home exam, the first one, and that's a protein, or sorry, a molecule that looks like double-stranded RNA. It blocks translation by phosphorylating EIF2, or leading to the phosphorylation of EIF2 alpha. So given what we know about poly-IC, and given what we know about HPV, sorry, I had to read back to the problem. So transfection of these cells with poly-IC will block translation of HPV. How? Or what allows for that to happen? So I'm going to give you guys a minute or two to kind of jot down a couple of notes for yourself. Given what you know about HPV and poly-IC, why would this block translation? Okay, so here is kind of just a run-of-the-mill basic explanation of poly-IC again. So poly-IC functions like double-stranded RNA. That's recognized by protein kinase R. Keep this in mind because we'll come back to this with Ebola as well. Okay, this pathway does become important. And PKR will lead to the phosphorylation of EIF2 alpha. That phosphorylated form prevents translation from initiating. The unphosphorylated form allows for translation to occur. Okay? When we say it inhibits or activates translation, what kind of translation are we talking about? It could be all translation. We will find some that have irises that can get around this block, but definitely cap-dependent translation would be blocked. Yeah. What's true about HPV mRNAs? Go ahead, Grace.
Can I guess?
Absolutely. That's the best way to do it.
So does E2F bind to the F?
So E2F will control host gene transcription. It won't control the viral transcription.
That was my guess.
That's okay. What do we know about? Let's start from the basic step of it. Andrew,
Doesn't it get translated kind of in the same mechanism the host DNA does?
it does. It does. It's translated in the same way that the host is. So what does that mean? So let's take a step back from that. What's true about the mRNA itself? What features does the viral mRNA have to have? Jonah?
Cap and tail.
Yeah, absolutely. What controls the transcription of HPV mRNAs? Or what actually performs the transcription process? That's the translation. What controls transcription? Polymerase, what kind? Host cells are DNA-dependent RNA polymerase. Since this is being driven by the host, that means the viral transcripts are going to look exactly the same as the host transcripts, or at least have the same basic features. They will have a 5' cap. We know that there's introns and exons, and that there's some alternative splicing that occurs before this mRNA is exported from the nucleus, and they're going to have poly A tails. And so because of these basic features, 5' UTR, there's our start. Because of these basic features, whatever blocks host translation would also block viral translation here. Okay. There's no modification done by the virus itself, other than the contributions of E2 to alternative splicing. But there's no other modifications to the RNA. And so it plays by the same rules. Okay. Questions on that concept? Annie?
Is that the same for all?
DNA viruses?
Yeah.
Largely, for many of them, yes, it is the same. It is the same. We've seen examples where this is different, though. So, like adenovirus, for example, has that ribosome shunting mechanism where it kind of by blocking the host cap-dependent translation, it allows for the viral transcripts to be specifically, and that's how it can overcome competition for the ribosome. We will see this mechanism in many, but there are exceptions, of course.
So what we've talked about before with the viral mRNA can like bypass all of the cap dependent, is that mostly with RNA viruses?
We see it more frequently with RNA viruses, or at least our examples of it are better with RNA viruses. So like poliovirus has an internal ribosome entry site, or an iris, that allows for it to bypass cap-dependent translation. So when that RNA genome is replicated, it's going to produce double-stranded RNA, which is going to shut down cap-dependent translation. The iris ensures that the virus still can replicate or translate its proteins. Yeah, great question. We've got 3 minutes. Let's just pick one of these proteins from the list. This, I think, might be the most comprehensive review question, the one that I have on here listed as number 2. So this is asking about what happens if the virus is defective in one of these proteins or one of these steps. So, for example, let's do the first one on there, actually, because this is a question that Grace kind of brought up. So you've isolated HPV with a mutation in one of the following proteins. List the step or steps of the viral life cycle that would directly be affected by the mutation, and explain the effect. So, for example, E2 disrupts an E2 mutation that inhibits its ability to bind to BRD4. Grace, what is this block?
Kind of like mitosis.
Yeah, when the cell is undergoing mitosis and ensuring that those viral genomes are going along with it, this prevents those viral genomes from being segregated to those daughter cells. And there's a couple of possible effects on this. One would be you get unequal partitioning. The second effect is what happens to DNA if it's not in the area where a nucleus reforms? It gets degraded. And so you can lose virus and viral genomes in that way too. If those viral genomes aren't segregated along with the chromosomes, well, when the nuclear membrane reforms, if it's hanging out in the plasmid, or if it's hanging out in the cytoplasm, it gets degraded. Okay? So we might say that we might not absolutely attribute this to a step other than saying like maybe biosynthesis, I suppose. Might be. It's not assembly. It's not maturation. It's not attachment entry or encoding. I think biosynthesis is where this probably fits best. And you should probably be able to do that for each of these on here. Okay, so I've got a couple others with E2, and then more broad ones with E6, E7, E1, E4, E5, L2, L1. Okay? Again, the major ones that I think you should be able to do, I'll just highlight which ones I think are probably most likely. E2s, E6 and E7 you should be able to do. I would say E1 you should be able to, because it is the helicase and that it should be fairly straightforward. E4 and E5 I'm less concerned with, though if you can figure those out, you're in really good shape. And then probably L1. And the effect of L1, L2 to a lesser degree might be similar. Okay, there are a couple of additional functions that we talked about with L2. All right. If you have any questions, let me know. I'll be grading the critical thinking over the next couple of days, and I'll put up a key in the next 24 hours. I realized this morning I had not created one yet. So I will put that up once it's done. Have a great rest of the day. See you guys in the next couple of days.